Assalamu alaikum. My name is Manahi Khanam Durrani and the topic of my today's presentation is phylogenetic analysis of an unknown DNA sequence. It is basically an assignment or task in which unknown DNA sequence was given. We obtained unknown nucleotide sequence from data file from teacher and we analyzed it on the basis of their phylogenetic relationships. So first of all, our first step was NCBI blast analysis. So copied all the data given in FASTA format, and you can see in this nucleotide blast, and click nucleotide blast after opening the NCBI blast analysis website. Next is on the next screen, enter the sequence query, paste the copied data. You can see the block in which enter query sequence option is given. Next is exclude model organisms and samples taken from the environment, while make sure database read nucleotide collection. Leave all the other settings as default and click blast on lower left corner. When results pop up, scroll down to the sequence producing significant alignment, while the high similarity sequences were downloaded in FASTA format. You can see that the sequences which have 100% similarity were downloaded. Only 10 sequences were downloaded in FASTA format. You can see that my sequence has similarity with Anabina species, NSC gene. Next is phylogenetic reconstruction. The sequence were aligned through cluster W and saved in MAGA format. Next is constructed a neighbor joining phylogenetic tree using MAGA 11 software based on the alignment of the query sequence with related sequences from different species. The tree revealed the evolutionary relationship between the query sequence and other cyanobacterial taxa. You can see that this is the aligned screenshot in which I tell you that how to align in MAGA X, and next was neighbor joining tree construction. This was a phylogenetic tree in which you can see that my query sequence was highly similar with Anabina species and a toxin A synthetase gene cluster complete sequence. This was the unrooted phylogenetic tree. So the evolutionary history was inferred using the maximum neighbor joining method. The evolutionary distance were computed using the maximum composite likelihood method and or in the units of the number of base substitutions per site. While the proportion of sites for at least one unambiguous base present in at least one sequence for each descendant clade is shown next to each other. While 12 nucleotide sequences were involved in this analysis, and this analysis indicated that provided DNA sequence originate from a cyanobacterial species with highest similarity observed with NSC gene and anatoxin A gene cluster of Anabina species and moderate similarity with Cuspodiothricus species. This was our result. And background information: cyanobacteria or blue-green algae basically are diverse photosynthetic prokaryotes found in various habitats. They play a vital role in global carbon and nitrogen cycle and oxygen production through photosynthesis, while Anabina, a genus of filamentous cyanobacteria, form long chain and heterocyst for nitrogen fixation, while some Anabina strains produce neurotoxin like anatoxin A, posing risk to aquatic life and human health, while gene cluster in cyanobacterial genome like anatoxin A synthase cluster associated with toxin synthesis. While Cuspodiothricus species and other filamentous cyanobacteria form bloom and produce hepatotoxin, while understanding genetic diversity and ecological role of cyanobacteria is crucial for managing harmful algal bloom. Cyanobacteria offer potential in biotechnology, producing biofuels and pharmaceuticals and aiding environmental remediation. Their role in carbon sequestration and nutrient cycling contributes to ecosystem health, while toxin-producing cyanobacterial bloom present challenges for water resource management and public health, emphasizing the need for comprehensive monitoring and management strategies. The next is function of NSC. NSC genes encodes and enzymes involved in the biosynthesis of anatoxin A, neurotoxin produced by Anabina species, while its mechanism is NSC gene enzyme catalyze specific chemical reactions that are essential for the synthesis of anatoxin A. The enzyme plays a key role in the assembly of anatoxin A molecule from precursor molecule, while importance in understanding the function of NSC gene provides insight into the molecular basis of anatoxin A production in Anabina species, while targeting NSC enzyme could offer potential strategies for controlling anatoxin A contamination in water bodies. That was mechanism of NSC gene. Next is Anatoxin A synthetase gene cluster in Anabina species. So genetic organization is basically the anatoxin A synthetase gene cluster consists of multiple genes including NSC involved in the biosynthesis of anatoxin A. While these genes are organized in a cluster often found on plasmid genomic island, regulation is expression of genes within the cluster is slightly regulated, influenced by environmental factors such as nutrient availability and light intensity. Implication is studying the gene cluster provide insight into the evolution and regulation of toxin production, while understanding the regulation of gene cluster could aid in the development of strategies that mitigate the risk of anatoxin contamination in aquatic environment. Next is regulation of anatoxin A synthetase gene cluster. So environmental influence is expression of the gene cluster is rightly regulated in response to environment use such as nutrient availability, light intensity, temperature. Regulatory mechanisms ensure optimal toxin production under favorable conditions, while adaptive response cyanobacteria modulate the expression of gene cluster to adapt changing environmental conditions and ecological niches. Regulatory elements within the cluster fine-tune the synthesis of anatoxin in response to environmental stimuli, while implication is understanding the regulatory mechanism provide insight into the ecological significance and potential ecological impacts of anatoxin A production. It informs strategies for monitoring and managing cyanobacterial blooms and associated toxins. And these toxins create contamination in aquatic ecosystems. So conclusion is that given the significant similarity with NSC gene and a toxin A synthetase gene cluster of Anabina species, the sequence may be associated with nitrogen fixation and toxin production, characteristics feature of Anabina cyanobacteria. Further investigation such as targeted sequences or morphological examination may be warranted to confirm the precise identity of the sequence and explore its ecological significance within the context of cyanobacterial diversity and toxin production. That was all from my side. Thank you for listening.